Okay, welcome to our section on blood. Let's see if I can get my cursor up and running here. Pointer options, laser pointer. Okay, here we go. So we have a diagram here. I sent that or posted it on my blackboard. You might want to print this out in advance. It might be a little easier to work with. This kind of describes, it's a real nice summary of our blood. So if we've got plasma, the stuff, the liquid of the blood and what's in that, and then the cellular elements, the blood cells there. If we spin things down in a centrifuge, all the cells go to the bottom, they become our cellular elements. Our plasma stays up top and there. This is for a human, we see here with the hand. For a cow, this number here would be closer to 65, or you could say 67, 2/3. Cellular elements, we'd be down around a third or 35, 33 moving forward. But pretty much what we're going to talk about here is all on this slide. It's a good summary. It's a good place to take notes if you want to print that out. Blood. Blood is a complex mixture. Very simply, it's all the constituents are suspended in a liquid. So your constituents are the blood cells, the solutes, the proteins, the hormones, everything that's in there. The solutes, concentrations, and the pH are very tightly controlled. Blood is held within very strict parameters, especially for pH. pH 7.4, slightly basic. If we vary more than a tenth of a point or 2/10 of a point in either direction, death is possible. So we're talking about very tight control here. In the, what is it, the hundreds place or the thousands place on a decimal. Those are really the only variation we get. And because those solutes are controlled, we maintain that osmolality, that isotonicness we've talked about. Depending on whether we include bicarb or not, we have 280 versus 300, with or without bicarb, looking at that. In a dairy cow, we're talking about the blood being about 7% of the body weight. So if you're looking at a 1,000-pound Jersey, you're looking at 70 pounds of blood. If you're looking at a 1,500-pound cow, you're looking at 100 pounds of blood in that cow. As we mentioned already on your diagram, about a third of the blood volume is cells. We can call that the hematocrit. What proportion is cells or packed cell volume? When we do that centrifuge that you see in that diagram, all the cells packed to one end, and then what percent of that linear tube is packed cell volume. 2/3 is solutes in solution. So with our plasma, we're going to talk about water that contains salts, proteins, and other stuff in there. So water, it's our solvent in the body. It's what dissolves everything. Plasma is 90% water, 10% dry matter for those of you that have had nutrition. So again, that is basically the vehicle. If we have 90% of 2/3, that's probably going to be more than 50% of the blood volume is water moving through, especially in our dairy cows. Salt. Sodium is the number one greatest amount that we're going to have. So you're looking at 140 to 150 milliequivalents per liter. That's a positive charged ion. Chloride, a negatively charged ion, is our second amount. Dairy cattle, you're probably looking at 90 to 100 milliequivalents per liter. Bicarb, we're down to about 20, 24 milliequivalents per liter. That's a negative charge. We got three positive charges there: potassium, calcium, and magnesium. Potassium, probably around 5%. Calcium, as we said, 2.5%, somewhere in there. and magnesium less than that. I don't happen to know that. So they're all present individual ions in solution. They are the salts of the blood. These salts serve three important functions. One, they help maintain that osmotic balance. They are dissolved in solution. They help maintain that isotonicness. So if you're looking at 140 for sodium and 90 for chloride, we're talking over half. The majority of the osmoles are coming from those two things there. So very important from that. They're going to serve as a buffer to maintain proper pH. The balance of positive and negative charges are going to determine pH, and the addition and subtraction of those ions by the kidney, by the large intestines, are going to help maintain that blood pH. And then we have salts are also going to regulate the membranes, particularly calcium, whether things can move in and out of the cell. So we need those ions to get those things done. Plasma proteins each has a specific function. Uh, Three specific ones we'll mention here. Albumin helps maintain osmotic balance, helps buffer the pH. The protein is going to contribute negative charges to the solution that are going to help balance things out. Albumin is usually a good indicator of nutritional status. And when we give an example of that osmotic balance, when we see kids from faraway countries that are starving in some way, shape, or form, they usually have a very skeletal head, very thin, bony arms and legs, but they usually have a big belly on them. And that's not because they're fat, it's because their water balance is off, and that's usually due to a lack of albumin. That's going to lead to more water around the intestines. Your osmotic balance is off. Fibrogen, that's the protein that helps form the clots that stop us from bleeding to death. You use the platelets, working with the fibrogen, the protein. You start that chain reaction to create that net, that web that catches the platelets, forms the clot. Immunoglobulins, also called antibodies, they are very present to help protect the body from disease. So once you've been exposed to something, we talk about this coronavirus, nobody's got any antibodies to it, but after you've had it, you're going to have antibodies, and that's what I think our governor today talked about measuring. If you have antibodies to the disease, that means you've had it, you've survived it, and we can send you back to work. Is what he Substances that we might find in the plasma, nutrients, carbohydrates, glucose is the big one. We got fatty acids, triglycerides from our lipids group, and we've got the proteins that we've mentioned before, the amino acids moving individually through the blood to where they're needed. You got waste products that are being taken to the kidney, to the liver, to the intestines to be removed from the body. You've got oxygen and carbon dioxide. They're sort of dissolved in the blood, not really. Oxygen is associated with the red blood cells. Carbon dioxide is kind of hidden in carbonic acid, bicarb in the blood. We've got hormones. We've got transporters that are carry things to protect the body in one way, shape, or form. Minerals, copper, and iron are very uh, nasty. They tend to rip things apart, so we put them in a package that allows them to be moved around the body and protects the body from those positive charges. They're rather aggressive. We also have our fats moving in carriers. These carriers, in both cases, are mostly protein, lipoproteins. In the case of fat, you've got proteins and fats together form a carrier. Uh, you hear on commercials the HDL, the LDL, and VLDL. The ratios of this can indicate your risk of heart disease. So you'll hear people bragging, well, I increased my HDL and I lowered my LDL, so I'm good to go. pay attention to those. The lipostatins will mention those in the background. Nobody knows what they mean normally, but you'll now know. So those are the plasma side. Again, we're looking at 65 here for dairy cattle. So that's the top part. We spin down. Now we'll talk about the 35% of that tube. The three types of cells are found in the blood. We have your erythrocytes, the red blood cells. They're pretty much purpose, and we'll talk a little bit more about that, moving oxygen around. That's their job. These are very, very, very tiny cells. In a cubic millimeter of blood, you'll have a millimeter, so you got something very, very small, millimeter by a millimeter by a millimeter. You have 7 million erythrocytes or red blood cells in that cubic millimeter. Leukocytes or white blood cells are there to provide immunity and defense for the cow, protect that animal from any outside threats or anything that is not the animal itself. In that same cubic millimeter, we're only going to have a few thousand, 7 to 10,000. If you look up here, your one of your suggested books there, Farm Animals. So red blood cell or white blood cells are much bigger. They're going to take up a lot more space, but they're intermixed there. So red blood cells vastly outnumbered. Leukocytes, our last thing. Platelets, they're going to work with fibrogen to clot the blood. So not quite nearly as many of those per. They're a lot smaller fragments, not nearly as many. Where do blood cells come from? We know we've mentioned already in class that they're made in the bone. Inside the bone, we have our factories. So you have a stem cell, kind of a generic, kind of omnipotent osteoprogenitor kind of cell that can become any of the three blood cell types. We have the white blood cells going through here. They come from two different sources. This source here also produces platelets, also produces the erythrocytes. Erythrocytes, or red blood cells. Like all the blood cells, they're going to originate in the bone marrow, that spongy bone area in the center of all our bones. That it's got a little extra space there, so the body says, "Hey, let's put it to good use." We make our blood cells there. Primary, or you could say the only purpose of the red blood cells is to deliver oxygen. We note that the red blood cells are very stripped down and very specialized. They're basically small discs of hemoglobin. The only thing that's left by the time they're manufactured, they take out everything. They take out the nucleus, they take out all the manufacturing, the lysosomes, the Golgi apparatus, the ribosomes. Everything is gone, more or less, from those cells except the ability to move oxygen. So carry it through the body and deliver it where it needs to go. When tissues of the body are not receiving adequate levels of oxygen, the manufacture of erythrocytes dramatically increases. So how does that happen? So some of you may have heard of the hormone erythropoietin. So if there's low oxygen in the tissues, the kidney secretes the hormone erythropoietin. So deep inside the body, the kidneys are sitting below at the bottom of your rib cage in the back. If they are sensing that there's not enough oxygen getting to the body, they're going to secrete erythropoietin, or we'll call it EPO. That's the kind of common name that you'll hear with people that are involved in doping scandals and things like that for athletics. Erythropoietin moves through the blood, goes to the blood marrow, and tells the blood marrow, hey, you need to ramp up the production of the red blood cells. and it's a positive feedback. The more erythropoietin you have, the more red blood cells are manufactured. Once you get back to a level of adequate oxygen, a few days down the road, the kidney stops producing that EPO. So it's always sensing, putting that out through there. So the system is turned off. Kidneys stop producing. They sense that there's enough. They'll stop producing the EPO, and the marrow eases off on the production of red blood cells. It's still going to produce them, but maybe just not at that accelerated rate. So the lifespan of erythrocyte is about 110 days for domestic animals in general. The lifespan of erythropoietin is less than 24 hours. So if we're going to produce a red blood cell, it's going to be around for more than 100 days. So we need to be careful of how many we produce at any one time. So once we increase the EPO level, it takes about 5 days for new blood cells to enter the circulation. You might note, this sounds slow, but it's a way to keep the checks and balances on the system. We don't want to overload the blood with red blood cells, or we make the blood too thick and it doesn't pump. You throw clots, it becomes a real mess. So we have a slow increase of red blood cells over time, and then EPO can respond very quickly. Yes, I have enough. No, I not. Kidneys can say, yes, we're got enough. No, we don't, and things can go back and forth pretty easy. If we have too many red blood cells, the too much EPO, we might find it difficult for blood to flow. So that actually becomes counterproductive. We can't get the oxygen and nutrients to the tissues because things are gunked up. So that slow release mechanism of EPO is beneficial to the animal. We don't have rapid increases and decreases in the red blood cells produced. You ramp it up and then you let it. Those of you that may have heard, I'm guessing most of you are too young to know this, but Lance Armstrong won 6 Tour de France's. It's a 2,000-mile bike race or something like that. 2,000, 3,000 miles around France. He was busted and stripped of his titles for the use of EPO and blood doping. So a lot of endurance people have been using EPO for a long time, and blood doping. Blood doping is when you take out your blood cells, red blood cells, and you put them back in just before the race, so you have an enhanced amount of blood cells in your body. So you get a big boost there when you put that back in. Both of those are illegal in most sporting events. So if we look closely at a red blood cell, we notice that it's not round or a sphere, but we have kind of basically two dishes on either side. So a donut without the hole. It's sort of, you took a jelly donut and sucked out the jelly, and it would collapse on itself. So they're very, very small, and this shape, it's known as a biconcave shape, allows the cell to have a lot of surface area for its volume. So if we had a sphere, you'd have as much as you could fit inside given the outside surface area. We want the surface area to be high. So if you do lots of little cells and you do this shape, the hemoglobin that carries the oxygen is always close to the surface. If you think about that shape that you see on the screen versus a sphere, the distance from the hemoglobin to the edge is somewhere in the edge of this uh, donut here. That's about as far away from the surface as the hemoglobin can get. If you made that a sphere, you could have 2 or 3 or 4 times that distance to an individual hemoglobin molecule. So lots of little ones in this shape allow the red blood cell to really do its job. This is a case where form follows function. So we called it a biconcave shape. This gives increased surface area per volume. This is a classic case of form following function. If we have the biconcave on very small cells, we generate lots of surface area, which provides more opportunity for gas exchange, for the oxygen to be collected at the lungs and released in the body as you go back um, through the circulatory system. In our erythrocytes, we have a molecule called hemoglobin. We have lots of them, millions and millions and millions and millions of hemoglobin molecules. So we have a protein molecule with iron at the heart. The iron is what binds an individual oxygen. So you have a one-to-one relationship between iron molecules or iron ions and the oxygen being connected. So this is a hemoglobin molecule. This is an example of tertiary structure. We have one, two, three, uh, four molecules of hemoglobin. We have beta chains that twist one way, alpha chains that twist another, and then you have a heme group in each of these. So this heme, this kind of squared off Saturn, is illustrated over here. Its structure. We have all these carbon atoms and then nitrogen atoms in the heart of it. This red dot in the middle here is our iron. Iron Fe is the symbol for iron, and that's what the oxygen sticks to. So each hemoglobin molecule here can carry one, two, three, four iron molecules or four oxygen. So how do hemoglobin in the erythrocytes handle the oxygen? It's really quite elegant and quite simple if you think about it. Um, Iron and oxygen have a natural stickiness to each other, especially at a neutral pH. So in the lungs, oxygen diffuses in, it goes down its concentration gradient. We'll talk about it more in the lungs when we get to the lung section. That oxygen gets close to a heme and then it sticks to that heme. It's going to leave the lungs, go to the heart, go out to the body, the capillaries. As it gets to a tissue that is actively metabolizing, that tissue is going to generate carbon dioxide. The carbon dioxide is going to drop the pH, make it a little bit more acidic, and that makes our iron a little less sticky. So tissues that are actively metabolizing, actively generating CO2, get rid of that stickiness and actually drop or make the oxygen come off where it's needed the most. Where a tissue that's working, metabolizing, using up that oxygen, that's where that oxygen is dropped off. So diffusion moves very quickly in and out of the blood vessel, and then the stickiness, very sticky to iron in the lungs, not as sticky to iron in the tissue. It's dropped off. If we don't have enough hemoglobin, if we don't have enough red blood cells, we get a condition called anemia. Anemia can happen anytime we either can't form hemoglobin, so that's a lack of protein, a lack of amino acids, a lack of iron, or we can't form red blood cells where there's a defect in the manufacturing process. We've got lost blood somehow, some way. Any of these conditions can lead to anemia. So if we look at what it's due to, we start with the nutritional deficiencies. Uh, Vitamin, mineral deficiencies are the most common thing, as I told you. For those of you that had me in nutrition, when all else fails for a deficiency sign, write anemia, because about 50% of the time you'll be right. Um, Minerals and vitamins, there's a variety of vitamins that are involved in the manufacture of red blood cells and their maintenance. Uh, Minerals, there's a number of different minerals that can lead to a deficiency, can lead to anemia. We have excess blood loss, some way we've got to bleed, we lose a lot. That's one way to lower our red blood cells. We have a defect in erythrocyte production, something wrong, something defective that leads to some sort of anemia. So, anywhere along the way, any of these things can lead to the same end result. So, that's why one of those things when you're figuring out anemia, there's a lot of things you have to check. In any case, we got low red blood cells, low hemoglobin. We don't get the oxygen to the tissues. We get out of breath faster. We don't have the endurance. All those things that we need oxygen for, we can't do. So we have our cells there. We're going to move on to the white blood cells. So 5 basic types. We'll get into the differences in a minute. These are what we see. We saw that 4 of those came from one branch in the bone marrow. One are developed on another. So overall, leukocytes are significantly larger than erythrocytes. These still have full function. They still have a nucleus. They still have the ability to produce proteins, antibodies, immunoglobulins. They have, the Golgi apparatus, the endoplasmic reticulum. They got lots of mitochondria. They are fully functioning cells that can move around and do things. The names for the leukocytes depend on how their appearance under stain. So, did they turn red? Did they turn blue? Did they not do anything at all? That'll help the names, and we'll show you that on a chart here in a minute. Um, Overall, as a group, they're responsible to the defense of the body. Each one or each type has a specific function, a specific part of the defense that they participate in. It's a great diagram here, or great table from the Clinical Anatomy and Physiology for Veterinary Technicians, second edition. So we have our types here, and then we've got 2 different lymphocytes, the B cell and the T cell. So macrophage, monocyte, those are kind of interchangeable. And then we have up here how they stain. So we've got things that stain. These do not stain down here. The eosinophil stains red. The basophil stains blue. The neutrophil, neutral, does not stain at all. The eosinophil, you'll hear that on commercials now. I lowered my eosinophil to cure my asthma. So if you listen to commercials, you might hear that. I think they're avoiding the use of white blood cells because if people say you're getting rid of their white blood cells, they might get upset. But you can see here in that commercial, it talks about asthma, anaphylaxis, allergic reactions. It's involved. If we have too many of those, things go overboard, so we need to reduce those. Phagocytosis, 1, 2, 3 of the white blood cells engulf whatever the problem is, whether that's a bacteria, a virus, splinter, something. They're going to eat it. They bring it into the lysosomes and they rip it apart. So kill it in some way, shape, or form. We did the eosinophil, the basophil. This is something that helps get things started. It helps identify to the body, kind of pointing out and saying, hey, there's a problem over here. You need to think about that. So they can get a reaction, or they'll invite the other white blood cells to the area and say, hey, you need to come over here. We've got a problem. Monocytes, we'll talk more about this. Antigens are what the immune system recognizes. So they might be a part of a cell, they might be a specific protein, they might be a specific virus. These are all antigens that the immune system responds to specifically. So monocytes are going to process those. They're also going to consume the problem. The B and T cells, our lymphocytes, are going to have 2 different modes of action. While these work in the body tissues, these might work more in the lymphoid tissues, moving through the lymph glands, things like that. You have two different modes of actions. One is you've got the B cells, they're kind of throwing things at an enemy. So you can think about it, archers, Spider-Man, something. They recognize a problem. This is our active immunity here, our specific immunity. They're going to produce antibodies and have a response that's going to attack a specific disease that they've seen before, they've learned, and they know how to act to it. And we'll talk more about that when we get to the immunity. We got the cytokine production and cell mediated immunity of our T cell. Our T cells more get a little bit more personal. They get up close to the problem and they secrete things that are going to destroy things. So you've got the superoxygen, you've got the hydrogen peroxide. They're going to basically take care of the problem kind of mano on mano, hand to hand. This is more of a distance thing. We're throwing things at the thing to tie them up, to hold them down so they can be consumed by the other um, white blood cells. One of the things we want to mention, going back to this diagram here, white blood cells, as you saw in the previous slide, they act in the body tissues. Most of the white blood cells are outside of the bloodstream. Bloodstream is just how they move around. They get in and out by going through the cracks in the capillary. So they'll work their way through, squeeze their way through to get out into the tissues. One of the initiations of the response, those cells there will start things. One of the things they do in a local zone is they'll loosen up the capillaries. So more white blood cells are attracted to the area that's initiating the cellular response, and then more will leave through the leaky capillaries. That's why we get swelling when we got a problem. And we'll talk more about that when we get to the immune response. So this is a good diagram here. We talk about the platelets. The there pretty much all they do is blood clotting. We're having somewhere between the 7 million that we associate with the cow, but more than the thing there. They're very, very tiny, so they're not going to take up much room. And that's, I guess, all we'll say about platelets. We'll have an assignment for you at some point to go through a chart to fill out to figure out who's where and help you study for your exam going through there. So let me write that down. Blood cell table assignment, or I forget. So I think that's it. Let's see if there's another slide.